All right, pre-AP biology. Today we're going to begin the second semester of biology. The second semester, Unit 5, begins with just sort of an introduction to DNA. We're going to talk a lot about DNA, a lot about genetics in this unit. So, kind of get us started here, I want to talk a little bit about um, a scientist who was pretty influential when it comes to understanding DNA. And his name was Frederick Griffith. And Frederick Griffith was a guy who studied bacteria. Um, specifically, one of the diseases that he studied was pneumonia. And one of the things that Griffith noticed about pneumonia bacteria is that there's two different types. There's what we call the virulent strain, or he called it the S strain. And the virulent strain of the pneumonia bacteria causes disease. So, for example, if you were to get virulent pneumonia, you would get sick. Okay, it would cause lung disease and whatever. Um, the other strain was a non-virulent type. He called this the R strain. And the R strain basically doesn't cause disease. So if you were to get this particular strain of pneumonia, you wouldn't get sick. It would just be in your body, but you would just fight it off really quickly and you'd be fine. So the big thing with Griffith was he didn't really quite understand why this was. Why was it that one type of pneumonia bacteria caused disease and then the other type did not? So he set up an experiment to try to see if he could try to figure out what was kind of happening here. So there's four different trials of this experiment. I'm going to kind of go through this sort of one at a time with you, and by the end, hopefully, we'll kind of get an understanding of what's going on. So what he basically did was, like most scientific experiments, that involve diseases, they're going to test them on mice. They're not going to test them on humans. That's morally, I guess, not the way to go about it. But so he took the S strain. This was the bacteria that caused the disease, and he injected that into a mouse or several mice, and the result was exactly what he thought would happen. The mice got sick, and the mice died. Okay, so not really anything surprising there in that first trial. Now, in the second trial, he took the R strain, which normally doesn't cause disease, and he injected that into the mice. And again, no surprise, the mice lived because the R strain doesn't cause disease. So the mice lived. Now, trial number 3 was kind of interesting. What he did in trial 3 was he took the S strain, that normally causes disease, and he killed it using heat. He just heated it up and basically killed the bacteria. So he injected that heat killed S strain into the mice, and again, he kind of expected that the mice would survive because the bacteria was dead, it couldn't cause disease, and that's exactly what he saw. So again, these first 3 trials are not anything out of the ordinary, right? S strain kills the mice, R strain doesn't. The heat killed S strain doesn't kill the mice either. Now, the fourth trial was the one that was kind of interesting. So, in the fourth trial, he combined two things. He took the heat killed S strain, right, that normally doesn't kill the mice. That's trial 3. And he added to it the live R strain. Now, the R strain doesn't kill the mice either. So when he injected those two things into the mouse died. And he thought this was kind of odd. He's like, well, why is it when these two things by themselves don't kill the mice, but then when you put them together in the same syringe and you inject them into the mice, the mice die? So, Griffith really kind of was, I guess, baffled as to what was sort of happening here. Now, eventually, what he figured out was that somehow these non-virulent bacteria acquired the ability to cause disease. and he didn't quite really know how this was happening. But he said that the non-virulent bacteria were taking something from the dead virulent bacteria that allowed them to sort of transform themselves into virulent bacteria. Now, eventually, what he sort of stumbled upon was something called conjugation. This is a process that bacteria can do. Other living things can't do this, but bacteria can. They can actually transfer DNA from one bacterial cell to another. If you look closely at this animation here, you can see that there's something kind of being swapped back and forth between the two cells. And this is something that, for example, like humans, we can't do this. We can't just, trade our DNA. Like let's say I have brown eyes and you have blue eyes. I mean, that's all caused by your DNA. I can't just take your DNA into my cells and get blue eyes. It just doesn't work that way. But bacteria can do this. They can actually kind of swap their DNA, and by doing that, they can actually trade genetic traits with one another. We call this conjugation, and it results in something called transformation, where the bacteria actually can take on different traits from what they normally have. So here's kind of a picture of this. Let's say, for example, down here, let's say this is your S strain. That was dead. It was like heat killed. And up here is your R strain, right? That normally doesn't cause disease. They actually can form this little bridge. It's called a conjugation bridge. It's almost like a little tunnel. And within that tunnel, they can actually swap DNA, or one bacteria can kind of take the DNA from another. So, what was happening here was the DNA that's in the S strain, that causes the disease. I'll go back to this real quick. Remember, the S strain causes disease. So let's say that this is the S strain here. So that DNA that's inside is what gives it its characteristics. Well, the R strain doesn't have that DNA doesn't have those traits. So through this process of conjugation, they can actually take the DNA from another bacterial cell, and by doing so, they can incorporate it into their own DNA, and they can transform themselves to have different traits. So that's kind of what was going on. I'm going to go back to this real quick. In this last trial, right? The R strain, which normally doesn't cause disease, was actually taking some of the DNA from the heat killed S strain. Just because the bacteria is dead doesn't mean that its DNA is completely useless, right? So, if the R strain bacteria took the S strain's DNA, then it now can cause disease. So that's sort of what was going on there. And Griffith, again, kind of stumbled upon this. Now, again, Griffith didn't really understand anything about DNA. He didn't really know what DNA was. He didn't really know the chemical makeup of it. But later on, a lot of other scientists kind of figured this out. I'm just going to kind of make a long story short, but DNA stands for deoxyribonucleic acid. Make sure we know this name. Okay, we should be able to recognize it. In your homework, you're going to have to be able to spell it correctly in the buzz reviews. So make sure we can sound it out, spell it correctly. Every living thing has DNA. All right, I don't care if it's a plant or a bacteria or a blue whale or a human, we all have it. It's the source of our genetic traits. So all of your genetic traits, your hair color, your eye color, your height, everything, skin tone, if you have any genetic diseases, I mean, that's all, you might remember we talked about this earlier in the year. DNA is what we call a nucleic acid. So it is an organic molecule. There are carbon-based compounds within it, but it is a nucleic acid, and nucleic acids are made up of building blocks called nucleotides. All right, so again, just a little bit of a review there. Now, also reviewing this, when it comes to a nucleotide, One of the things that I kind of want you to know is the chemical structure of it. All right, every nucleotide, and there are some different types, which we'll talk about in a minute, but they all have sort of the same three general components to it. All right, one of them is what we call a phosphate group. All right, it's PO4. That's what we see down here, right? You have a phosphorus atom bonded to four different oxygens. So that phosphate is always a component of a nucleotide, okay? The second part is a sugar, right? Here's our sugar unit here, right? Carbon, hydrogen, oxygen. This doesn't show you every single atom, but anyway, the sugar's name is deoxyribose. That's where the deoxyribo comes from in DNA, but deoxyribose is the name of the sugar. So there's a sugar unit as well in a nucleotide. And then the last thing is something called a nitrogen base. Now, the nitrogen base is always attached to the sugar. So you'll notice here that you have the phosphate attached to the sugar, and then on the other side, you have the nitrogen base attached to the sugar as well. Now, as far as these nitrogen bases go, there are four different types. There's like four different flavors of bases. All right, we call them guanine, cytosine, adenine, and thymine. So, like for example, this picture here showing you this piece of DNA. You'll notice all these different colors, right? These different colors are showing you just the different nitrogen bases that make up the molecule. All right, but we need to make sure we know these names: guanine, cytosine, adenine, and thymine. Now, a little bit more to this. These bases, um, all four of them, we can kind of put them into two different categories. We call them purines and pyrimidines. And the big difference between the two of them is just kind of the molecular shape of them. There's a little bit of chemistry involved, and I'm not going to get into that too much with you, but the purines have a distinctive shape. All right, now the two purines are adenine and guanine. You just have to kind of know that. The pyrimidines are thymine and cytosine. They have their own like distinctive shape as well. Now, one thing about DNA, and this is called Chargaff's rules. Some guy named Chargaff figured this out back in 1949. Is the amount of adenine that you have in a DNA molecule is always equal to the amount of thymine. And the reason why that is because adenine and thymine bond together always. They're always connected together. Guanine and cytosine, the same thing. The amount of guanine and the amount of cytosine is always the same. So, for example, in a DNA molecule, if let's say 20% of the DNA is made up of guanine, well, that means that 20% is also made up of cytosine, because these two things are always found together. They're always found in the same amounts. So adenine with thymine, guanine with cytosine, that's sort of the rules of Chargaff or Chargaff's rules. All right, this picture here kind of shows us more of a kind of a layout of what DNA kind of looks like, kind of the structure of it. And what I want you to look at down here is this box that I kind of highlighted down here, right? So what we're seeing is a nucleotide, right? You've got your phosphate group here. Here's your phosphate. Here's your sugar, right? This is your deoxyribose. And then here's your base, right? There's four different bases. This one happens to be guanine. So if you have a phosphate, a sugar, and a base, that is a nucleotide. And you'll notice right above it, there's another one, right? There's a phosphate, there's a sugar, there's a base, right? Right above that, there's a phosphate, a sugar, and a base. So what we have here on the left-hand side is this big, long chain of nucleotides. That's all it is. Now, the only difference between each nucleotide is there's maybe a different base, okay? But for the most part, their structure is the same. Now, on the other side, you'll notice that there's another chain of nucleotides. So again, we have a phosphate, a sugar, and a base, right? Phosphate, sugar, base, phosphate, sugar, base. So, the sides of it, it kind of looks like a ladder. This is kind of the steps of your ladder in the middle. Then on the sides of the ladder, this is sort of the sides of it over here on the left and the right. But you'll notice in the middle, you've got, for example, if you have guanine on this side, right, you've got cytosine on this side. Anytime you see a G, you also see a C, right? So G with C, C with G. Here we have adenine, right? It's always paired up with thymine. Thymine with adenine, guanine with cytosine, adenine with thymine. And these bases are all connected by bonds. These are called hydrogen bonds. We've talked about that briefly before. So essentially what you have is a chain of nucleotides on one side, a chain of nucleotides on the other side, which is sort of the opposite chain, and they're all connected in the middle with these hydrogen bonds. All right, so this is what DNA is. This is the chemical makeup of it. We need to be familiar with this structure, okay? So you need to study it. You need to learn it. You need to memorize it. All right, go to a tattoo parlor and get it tattooed on your arm. Whatever you have to do, you have to learn this. All right, a little bit more with DNA. Watson and Crick. You may have heard of these guys before. James Watson and Francis Crick are credited with the discovery of the shape of a DNA molecule. DNA, even though I kind of said before, it kind of looks like a ladder. You've got like the steps, the rungs of your ladder in the middle. You got the sides of your ladder over here. It actually is a ladder, but it's twisted. So we have a special name for that. It's called a double helix. A double helix is kind of like a spiral staircase looking thing. But again, you've got like one side of nucleotides, and on the other side, you have the exact opposite. So again, you can see here in this molecule, you see like the different colors, right? It's not really colored in real life, but like this little orange base here, and then you've got this kind of purple base over here. So if this is cytosine, then this is guanine, right? Cytosine, guanine. Then you have your two different colors here, right? So you've got a thymine, let's say, and then adenine. So everywhere you see silver, you're also going to see green, right? Here's green and there's silver with it, right? There's green, there's silver with it. You see that all the way around. Anywhere you see orange, you're going to see purple, right? That's because cytosine always bonds to guanine. So there's a reason why it looks like this. It all has to do with those Chargaff's rules. And of course, it's sort of that twisted ladder shape that we're probably somewhat familiar with. All right, one more thing or a couple more things here. And again, I just want to sort of connect this to what we talked about last semester. DNA is found on chromosomes, right? We learned last semester that we have 46 chromosomes in all of our body cells, except for sperm and egg, right? We have 23. So, these chromosomes here, here's an example of some here. These things are made of this. This is what they're made of. Okay, so all of your DNA is found on those 46 chromosomes. So if we were to take one of your chromosomes and unravel it, you would basically see this structure here, right? This is what it would sort of look like. And I want to kind of show you this. I think this is kind of a picture that sort of summarizes really everything we've talked about the last couple of weeks, right? Here's your cell. Let's say this is one of your skin cells or blood cells or whatever, right? Inside the nucleus there, you've got your chromosomes. Right? There's 46 of those. There's not 3, but whatever. And each one of those chromosomes, again, if you unravel it, you'll have all of your DNA. All right. Now, there's a term here. Here's a term we haven't used yet. It's called a gene. A gene is a section of DNA that codes for a particular protein. Remember what cells do, right? They build proteins, but cells need the instructions to build a protein. Well, the instructions are on your DNA. All of these little letters, T and C and G and A and all this stuff, all they are codes. And they basically are instructions for your cells to build proteins. So each gene is a set of instructions to build something very specific. So if you have those instructions, then your cells can make proteins. If you don't have those instructions, or the instructions are mutated or they're incorrect, then your cells can't make those proteins. Okay, so that's sort of the big picture of all of this. All right, guys, I'm going to leave it here for today. I will see you soon.